Hi, and welcome to this lecture on environmental adaptation of animals. This is the second video combining development biology on the environmental adaptation, and it's titled Epigenetics. Well, even in human development, the environment is affecting. And this can be demonstrated easily by comparing the height of children today on the children that were born 150 years ago. So the children today, they are taller, they start periods a little bit earlier, they live longer. And still the human genome is not drastically changed during the last century. This means that the development is not based only on the instructions written in genes. And how this environment is affecting on development, it's been discussed largely on our bachelor level course Development Biology, but perhaps you have not participated on that course, or perhaps there is a need for short review. Well, first of all, the environment can affect on your sex. So in several ectothermic species, the temperature determines whether the embryos will turn male or female. Like in Mississippi alligator, if the eggs are in 32 or 33 degrees of Celsius, they all become males. If it's hotter or colder, they will become females. And this can maximize the number of females in the population. And if you have maximizing the number of females, then you are maximizing the offspring. But on the other hand, it also limits the temperature zone in which these species can live. So if it's too cold or too hot, you will lose one sex, and then you lose whole population. And actually it's been speculated that this has been one of the problems in the dinosaurs. So when the temperature was changing, they lose first males or females, and then they lost completely the species. Also, the predators may affect on the phenotype. On these two marine mollusks, they synthesize thicker shell in presence of a predator, meaning a crab. So the shell is only 10% thicker, but it sends some chemical from the crab to start this synthesizing. And also, we can have quite tricky phenotypes. These butterflies look completely different, but still they are the same species. It's a western white butterfly that has two different phenotypes. So there is one for spring generation and one for summer generation. And the regulation is done by day length and the temperature. Well, it's been actually speculated that in the spring generation, why it's dark wings, is because it's absorbing as much heat as possible to get the butterfly flying. And how does this butterfly shift this phenotype is based on two hormones, juvelin hormone and ecdysone hormone. And this might be the most extreme example of changing the phenotype. It's a locust that has two different phenotypes, and they switch when the animals are interacting with other animals. So it's meaning that, okay, the population density is quite high when they are facing the same species. And that affects that they are changing for the migration phenotype, and these migrating phenotype form swarms that are eating all the vegetation in large areas. And in this locust, only one neurotransmitter is needed for switching the phenotype. There's also another migrating locust that has, beside serotonin, also dopamine, that is also a neurotransmitter that has a main role in this switch. To understand how we can change these phenotypes, we must understand something on the DNA and its modifications. Well, the DNA contains all the genes, so all the instructions for all the proteins. And this DNA can be modified by adding methyl groups. And so this methyl group adding, it's called DNA methylation. And the tricky part of this methylation is that when you are methylating the DNA, it will be copied in the offspring of your cell. So in the next generation of the cells, of this cell, they will also be methylated. And the methylation is quite often in the news, and it's said that, okay, it's either bad thing or good thing, but it's actually a very natural thing. So we have one nucleotide, cytosine, in which this methylation can happen. So we can methylate, but we can also demethylate it. And in human development, we are methylating the gamma globin gene in the early development. So it's the promoter area which is needed to have this gene active. It's methylated, so it's inactive. And at the same time, the epsilon globin is active because that promoter area is unmethylated. But a little bit later during the development, this epsilon globin is methylated, so it's inactive, and this gamma globin gene is now activated. So that's why the fetus has gamma globin, and if you remember, the gamma globin was part of the fetus type hemoglobin that was having high affinity for the oxygen and stealing the oxygen from the mother. Also, the food can be triggering. In several social insects, they determinate, for example, in honeybees, when the embryo will be queen or worker. And quite often in these social insects, it's been the switch has been done by methylation and packing the DNA. And this is one example, Florida carpenter, that is switching between the major and minor type of, and it's depend on the packing of DNA. It's called histone acetylation. And it can be tested by manipulating the packing. It switch the major and to act like a minor, so it's gathering more food for the society. And these kind of methylations are very typical, found nowadays also in human population, how food is affecting and how are certain kind of environmental risks, etc., are affecting on the methylation, etc. So it's getting quite popular nowadays. Thank you.